first video on nucleic acid structure is going to be pretty short because I'm going to assume that you've already watched or know about structures of carbohydrates, fats as triglycerides, and then amino acids that are polymerized into proteins. And so I'm going to move on now and just talk about the structure of nucleic acids and importantly highlight that this is sort of one thing that is not like these other three. Carbohydrates, fats, and proteins are all macronutrients that we use either as building blocks or fuel or a combination of both. Nucleic acids really are not intended to be fuel, and so we're not going to have a number that we think about in terms of their caloric value. We're also not going to have a location of where we have reserves like we do for some of these other molecules. And importantly, we're really not going to talk about, an important physiological concentration. Nonetheless, it is important to think about not only how we biosynthesize the amino acids or the nucleic acids that we need, but how we sort of degrade nucleic acids and then those corresponding nucleotides. Importantly, we'll have kind of two stories that we'll sort of talk about here. One has to do with the degradation of purine nucleotides and how that leads to the disease gout. And then we'll also talk about sort of the ability to hijack some of the machinery to make nucleotides, specifically deoxy-T, and that is a very potent chemotherapy drug. Okay, so a little bit here. I'm just going to zoom in. I'm not going to really go through the details of structures or anything that we have over here. Just had a high-level kind of discussion just a second ago there. But if you need some more, there is a video that goes through all three of these structures. So I'm going to remind us, these are all things that I want you to still be able to do. Draw these basic structures. If we were to draw a little dinucleotide, we're going to have our 5' end, and then we're going to have our ribose sugar. Okay, so don't need you to know the structure of the bases. I want you to distinguish whether it's a purine or a pyrimidine sort of nucleobase, but not specifically drawing or recognizing A's and G's versus T's and U's. Do want you to know who they base pair to though, okay? But this is again the 5' end, okay? And at this 5' end, we can have a hydroxyl group. As we'll see in a minute, we could also have a phosphate. So nucleotides are connected together by a phosphodiester bond. So again, here's our phosphodiester bond. And once we get here, then we're ready to repeat our sort of structural repeat there. And we've got a base. So a couple of things to highlight here. Down here, we're going to have an OH group that can be our 3'. So we've got directionality here going sort of 5' to 3' in terms of when we think about directionality of this biopolymer. We do want to highlight that we can have additional phosphates that can be added either onto the 5' hydroxyl group or the 3' hydroxyl group. So that can be an additional way to sort of functionally modify. And importantly, the sugar that we have here can either be a hydrogen or an OH group at this 2' position. And so this 2' hydroxyl is going to be what we see in ribonucleotides or ribonucleic acids. And then when we have as a hydrogen, we're going to see that's when we have our DNA flavor, our deoxyribonucleotides or deoxyribonucleic acids. So again, and we tend not to see biologically where they might be mixed, where we have what we call a chimera, where it's sort of a mix of deoxy and ribo sort of situations. But we do have both of those flavors that we see sort of biologically, and we'll talk a little bit more about those in another video. So beyond drawing sort of our basic nucleic acid here, so again, this is a nucleic acid, and a nucleic acid is really polymerized nucleotides. We really didn't talk about sort of, and we're not going to talk about here, sort of the really neat biochemistry that has to do with formation of phosphodiester bonds, right? We talked about RNase A and we talked about the hydrolysis of phosphodiester bonds, but we're really not going to talk too much about the synthesis of nucleic acids, or for that matter, the mechanisms for the synthesis of proteins. Really neat sort of things, but hopefully that's something you get in a genetics course. So when we think about nucleotides, I want you to remember that that T reminds us that there's going to be 3 pieces that we have here on a nucleotide. We're going to have the nucleobase, we're going to have the sugar, and then we're going to have between 1 and 3 phosphates. Okay, this again, if it's just the base, it's just called a nucleobase. It's called a nucleoside if it's with the sugar, and that S can help you remember that piece, and then 1 to 3 phosphates are going to be appended on there to generate the nucleotide, either mono, di, or triphosphate. So a couple of other things to highlight just as we sort of wrap up this intro video here. It's important to highlight that we're really not considering nucleic acids or their corresponding monomeric nucleotides as really being fuels. They're not really macronutrients in the way that carbohydrates, lipids, and proteins, as things like glucose and triglycerides and fatty acids and then amino acids really are. But one other thing to highlight is that they are also not essential. And remember what that word essential means. Essential means that you cannot produce it yourself, so you need to consume it from your diet. So it's not to say that nucleic acids and nucleotides and all of that are not important. It just means that we can make everything that we need with the biochemical machinery that we have in our body. So last little bit to think about, and we had in separate slides to kind of address this when we did our lipid and then our protein metabolism, but we do have both endogenous and exogenous sources that we're going to have of nucleic acids and corresponding nucleotides. So from an endogenous standpoint, anytime we do cellular turnover, we are going to be degrading those components. So we can sort of have this internal sort of source of nucleic acids that we need to think about breaking down to nucleotides and then degrading those nucleotides or correspondingly building them up. But we're going to focus a little bit more on this exogenous piece. And the key is that all food comes from living organisms, and those living organisms have cells. Those cells have nuclei, the nuclei have DNA, and so there are nucleic acids that come along with any food product. Now, one of the things we'll talk about as we talk about gout is that there's some food products that are more enriched in nucleic acid than others, and so those are going to be more problematic for somebody who has a problem metabolizing nucleic acids. Okay, so all foods have DNA because they come from living organisms. So, little brief overview, thinking about the structure that we have for nucleic acids and then nucleotides, and then again, not too much to think about like we had for our other macronutrients because nucleic acids really are not a fuel and they're not really essential. So we don't need to think about sort of maintaining certain physiological concentrations or consuming them in our diet.